Myself, Dr. Raktim Mukherjee, Assistant Professor and Head of the Department, Shri P.M. Patel Institute of PG Studies and Research in Science, affiliated to Sardar Patel University, Anand. In this second video of the series, the title of my talk, though it's epilepsy, I shall focus on astrogliosis. And before I come to astrogliosis and explain a phenomenon where lot many research papers have been introduced, and to have a basic understanding on the phenomenon of astrogliosis, I will focus before you a research paper from Frontiers in Neurology. A part of the lecture I have taken from this. The title of the paper speaks Astroglial glutamine synthetase and the pathogenesis of mesial temporal lobe epilepsy. This review paper gives a very good articulation to the fact that astrogliosis is indeed one of the reasons behind development of epilepsy. But at that moment we do not know anything about astrogliosis. So, let us focus on astrogliosis. So, the next part of my talk will be what exactly is astrogliosis. So, what is astrogliosis? You all might be aware that neurons are never alone. Neurons are the structural and the functional unit of nervous system. Neurons are always accompanied by extraneuronal cells. These extraneuronal cells are also called as glial cells. And there are 5 types of glial cells: oligodendrocytes, ependymal cells, astrocytes, the Schwann cells, and the microglial cells. So, total there are 5 glial cells. It is always said in neurology that for every single neuron there are 10 raised to 10 extraneuronal cells. So, when I talk about astrogliosis, essentially we refer to astroglial cells or astrocytes. So, astrogliosis is also known as astrocytosis. Why it is called as astrocytosis? It is called as astrocytosis, or it is also referred to as reactive astrogliosis. All these terms you will see in research papers. What exactly is it? It is abnormal increase, not a normal increase. It is an abnormal increase in the number of astrocytes, and this number of astrocytes that increases can be due to various reasons. One of the major reason is destruction of the nearby neurons. And what are the reasons why nearby neurons are destroyed? And these nearby neurons are from central nervous system. They are destroyed because of trauma. What kind of trauma? The trauma can be infection, that is neural infection or glial cell infections or brain infections, ischemia, we already know, temporary cessation of oxygen supply to the parts of the brain, stroke. We have already introduced ourselves to ischemic and hemorrhagic stroke in introduction. We will be studying stroke in detail as a topic. Autoimmune response to neurodegenerative disease. Neurodegenerative disease. Neuro refers to the nervous system. Degeneration is when there is a wasting away or death of neurons, certain types of neurons, that is called as neurodegenerative disease. Astrogliosis in the temporal lobe epilepsy. What is astrogliosis in the temporal lobe epilepsy? Astrogliosis in temporal lobe epilepsy, first of all we have to understand that there are various lobes of the brain, and to understand temporal lobe, first as a layman we should understand the different lobes of the brain. So, temporal lobe epilepsy are characterized by astrogliosis, and this is your temporal lobe, the location of your temporal lobe. To continue, my dear students, this is the temporal lobe, the lobe which is responsible for astrogliosis. Apart from that, you should be in a position to know other lobes of the brain. This is the frontal lobe, that is the parietal lobes, and this is the occipital lobe, the cerebellum, the balance center of the brain, and this is the brain stem. So, in short, there are 4 major lobes of the brain: temporal lobe, frontal lobe, parietal lobes, and occipital lobes. When you talk about astrogliosis, the microglial response leads to increased proliferation, more number of cell division, increased proliferation, and hypertrophy. Hypertrophy is increase in the mass or size. So, hypertrophy of astrocytes. So, more number of astrocytes, that is astrogliosis. Now, in a very fantastic flow chart fashion, I have covered astrogliosis so that it becomes easy for my students to understand. What are the changes related to astrogliosis? Astrogliosis in turn are associated with changes in the membrane conductance, how the electrical impulses of the brain are conducted, and this affects the membrane conductance for ions. How it is affected? It limits the capacity of astrocytes for the following ions: potassium K plus, calcium Ca2+, and hydrogen ion, that is H plus. So, they limit the buffering capacity of astrocytes for all these ions. Because of the limiting of the astrocytes, what exactly happens? That there is a disruption in homeostasis. Once the homeostasis is disrupted, there will be 2 major changes. One, there will be changes in neurotransmitter glutamate, and there will be changes in neuromodulators such as adenosine. So, this is the simple straightforward flowchart way of explanation, changes related to astrogliosis. Someone can ask you in exam, what are the changes related to astrogliosis? These are the changes. This is the diagram from your study material. You have the excitatory presynaptic neuron, you have adenosine, you have the reactive astrocytes, astrogliosis, and this diagram tells you about the various neurotransmitters involved. You have an excitatory presynaptic neuron and an inhibitory presynaptic neuron. Whether it is excitatory or inhibitory, the effects are going to be different. You have the sodium channel which is upregulated, the potassium channel is downregulated. Likewise, in inhibitory presynaptic neuron, the GABA neurotransmitter is upregulated. So, what we will do, this diagram we will see in the form of flow chart for better understanding. So, this is what I try to explain. What are the consequences of altered astroglial physiology? The extracellular concentration of potassium and glutamate are increased, whereas that of adenosine is decreased. So, you can always see the flow chart mentions that adenosine is decreased, whereas glutamate and potassium increases. So, extracellular concentration of potassium, I have tried to show extracellular by showing it outside this boundary of the cell, extracellular. So, what happens? Extracellular potassium and extracellular glutamate, there is an increase. There is an increase in extracellular potassium and glutamate, whereas adenosine is decreased. The nucleotide adenosine is decreased. Why there is adenosine deficiency in epilepsy? That is interesting. We simply cannot escape at master's level that why adenosine should decrease. We should know the reason. Why adenosine decreases? So, adenosine decreases. To understand adenosine decrease, we have to understand the transmethylation pathway. These are the key biochemistry, because your subject is medical biochemistry. So, these parts are really important. So, what is this transmethylation pathway? We try to understand that transmethylation pathway. There is a difference in the physiological transmethylation pathway and the pathological transmethylation pathway. Physiological transmethylation pathway, you have S-adenosine adenyl methine SAM. Under the influence of methyl transferase, that is DNMT, you have S-adenosyl homocysteine. Under the influence of SAH hydroxylase, the adenosine in the presence of adenosine kinase gets converted to AMP. Whereas under pathological condition, the methyl transferases leads to methylated DNA. Methylated DNA leads to adenosine getting converted to adenosine kinase, and this pathological transformation is detrimental, and that leads to deficiency. So, what is the role? There is an overexpression of adenosine kinase. Adenosine kinase is overexpressed. This adenosine kinase is also the key enzyme responsible for clearance of adenosine from astrocytes. So, what it implies? It implies more adenosine kinase, the more will be clearance of adenosine. And it is a metabolic clearance of adenosine. So, obviously, when there is overexpression of adenosine, the metabolic clearance of adenosine would take place, and automatically any molecule that gets metabolically cleared, that molecule will always fall short or there will be deficiency. So, adenosine kinase has been identified as the key pathological hallmark in epilepsy. It is because of the overactivity of adenosine kinase. So, what adenosine kinase does? Adenosine kinase converts adenosine into AMP. So, automatically more adenosine kinase means more AMP. More AMP means less adenosine, right. So, this is how there is deficiency of adenosine kinase. Once again I explain, because it may be asked in examination, you have to understand why there is a deficiency of adenosine in epileptic patients. Reason: it is attributed to the activity of the enzyme adenosine kinase. This enzyme adenosine kinase is overactive. What this adenosine kinase does? Adenosine kinase converts adenosine into adenosine monophosphate AMP. More adenosine, more enzyme means more conversion of adenosine to adenosine monophosphate or AMP, and therefore, all the adenosine is converted to AMP. Therefore, the concentration of adenosine drastically falls. Therefore, we have adenosine deficiency in epilepsy. This adenosine is also called as retaliatory metabolite. Retaliation. Can anyone tell me what is retaliation? What is retaliation? Quick. What is retaliation? Should I give an example? Recently, Hamas militant organization of Palestine initially hurled so many rockets at Israel. What did Israel do? What did Israel do?
Counter attack.
Yes, the counter attack is called as retaliation. You hit me, I will hit you back. That is retaliation. So, this adenosine is also called as retaliatory metabolite. Why it is called as a retaliatory metabolite? Because this molecule adenosine is a key link. It is a key link between energy homeostasis and metabolic activity. This metabolic activity and energy homeostasis, the metabolic activity has been termed as retaliatory metabolite. Retaliatory metabolite. Why it is called as retaliatory metabolite? Because of the capacity to adjust energy consumption to energy supplies. How much energy has to be consumed based on the energy supply, just like your multi-point fuel injection system of your latest car engine. This molecule is also called as a retaliatory metabolite because of its capacity to adjust energy consumption to energy supplies. So, I have just highlighted that point, that adenosine is a connecting link between energy homeostasis and metabolic activity. This is the reason adenosine is termed as a retaliatory metabolite, due to its capacity to adjust energy consumption to energy supplies. Now, based on this, there are lots of research paper, and one I have taken from popular newbies research paper from very prestigious journal, Trends in Biochemical Sciences. So, whatever I am going to teach the next segment, this is from the research paper, Adenosine and the Concept of Retaliatory Metabolite. This is from Andrew C. Newboy from Trends in Biochemical Sciences. You can see the research paper deals with some of the popular biochemical steps, because it was in 1970s Satin and Val reported adenosine elevated cyclic AMP concentration in the cerebral cortex sizes. That is how adenosine can modulate a wide array of physiological process, including our today's topic, that is epilepsy. Another research paper was from Freedom in 2007, Cell Death and Differentiation, that adenosine is an endogenous distress signal and this modulates tissue damage and repair. Students, one of the reason why I am giving screenshot of the various research papers, two reasons. Any student who are ambitious readers, want to build their career, they can always go through this research paper as extra reading. Second, students can be aware which is the source from which I have taken my lectures or some of the slides. And third, I want to convey in a very candid tone that student, whatever we are studying, the role of adenosine is very key in epilepsy. It has lots of biochemistry involved. So, this is the diagram. You have outside the cell extracellular and inside the cell intracellular. You have adenosine. Adenosine has four type of receptors: adenosine receptor A1, adenosine receptor A3, A2B and A2A. Whereas calcium enters through calcium channel, you have linked with protein kinase, potassium leaks out of the cell to potassium leak channel, and you have the G0 and the G1 subunits of G protein. And adenylate cyclase, always called as membrane bound adenylate cyclase, converts the ATP to AMP, okay, cyclic AMP, ATP to cyclic AMP, and cyclic AMP in turn activates protein kinase. So, this is the story so far. What we are going to study, that presynaptic glutamatergic terminal is there. You have A1R and A1R receptor, adenosine receptor, and you have plus sign, and there you have the, what happens is during stress, what is the role of stress and adenosine? During stress, adenosine rises. Adenosine rises during stress. Any type of stress, adenosine rises. And this adenosine rise affects the neurons. How it affects the neurons? Once there is stress, the neurons become active and it further limits neuronal energy consumption. It further limits or decreases the neuronal energy consumption. How it does? You have adenosine, very important, listen very carefully. Adenosine, there is adenosine. Adenosine binds to adenosine 1 receptor. This in turn has to roll with inhibitory regulatory G protein, that is GI. GI is inhibitory. First semester we have studied. GI inhibits adenylate cyclase. Once adenylate cyclase is inhibited, what adenylate cyclase does? Students, quick, unmute and speak. What adenylate cyclase does? Catalyzes the conversion of
ATP to AMP.
ATP to cyclic AMP, right? So, it inhibits adenylate cyclase. Adenylate cyclase normally converts ATP to cyclic AMP and 1 phosphate, but it gets inhibited. Once adenylate cyclase gets inhibited, two important aspects happen. One, it augments the neuronal hyperpolarization. How it augments? It augments the G protein gated inwardly rectifying potassium, that is called as abbreviated as GERK. So, this is your G protein gated inwardly rectifying potassium, that is GERK, and this augmentation leads to neuronal hyperpolarization. On the other hand, it also affects the presynaptic neuron. In the presynaptic neuron, it blocks calcium channel. So, therefore, calcium cannot enter the presynaptic neuron. What are the energetics of epileptic seizures? Because in epilepsy, electrical alterations of the brain or epileptic seizure is the main phenomenon. So, in epilepsy, seizures consume large amount of energy because there is an alteration. So, lots of energy is consumed, and once energy is consumed, this leads to rise in adenosine, because adenosine acts as an endogenous, that is within the cell, anticonvulsant and seizure terminator, naturally acting endogenous anticonvulsant and seizure terminator. So, that concludes the segment on astrogliosis and epileptic